Okay, everybody, welcome to your next lesson on plasma membranes. So just to give you a bit of a heads up, a plasma membrane, it is synonymous to the cell membrane. So what we're going to be talking about today is really just the cell membrane. Now, the thing is that this actually will apply to like any membrane at that point. So, when you think about what things have membranes, we always talk about like the cell membrane, the plasma membrane, right? So here's your cell, right? You have like the nucleus and, all your organelles and so this is going to be a membrane, right? But then like if this is like your mitochondria right here. Keep in mind your mitochondria also has membranes as well. Your nucleus, right? The nucleus has the nuclear envelope. That is also a a plasma membrane. So we're basically talking about all these different membranes that exist. Now, specifically in AP Bio, we're really going to be focusing on specifically the cell membrane, plasma membrane. But what you can do is use any of this information to kind of describe anything that we end up seeing going forward. So the big thing that we're going to really need to go take a look at is going to be this guy right here about the phospholipids. Now, just to remind you, the phospholipids, we kind of described them a little bit back in biochemistry. It looked like the head with little dangly tails where you had your polar head and then you had your non polar tail, right? And they're going to orient themselves in a way. We're going to go show you that momentarily. All right, now inside of your membrane, or I guess like kind of traversing it, you're going to have these embedded proteins. And now these embedded proteins will have specific intermolecular forces that will explain how they're going to interact with the cell membrane. Some of them might be hydrophobic, some of them might have a charged or polar side. Some of them might have some other stuff that's going on. So what we'll do is we'll kind of link back to our intermolecular forces, right? Our IMFs. And we'll be explaining why the proteins are set up the way they are. And then we can also kind of describe if we change the conditions on the solution or chemicals that are nearby where the cell is, it can end up affecting how that protein is folding, right? Because we kind of discussed that a little bit when we talked about how like your amino acid chain can end up becoming denatured, right? So then potentially that can end up affecting the overall function of your proteins that are in your cell membrane. So like maybe antigens or transport proteins aren't working properly. And then finally, this is going to be talking about like the part of the actual cell membrane right here, which is your phospholipid bilayer. You got proteins, steroids, specifically cholesterol that's embedded within there, and then you got these glycoproteins and glycolipids that are used in order to go and have your cells basically talk to each other, for lack of a better term. Right. So this right here, the blast from the past, right? You probably did cell membranes, right? Cell membranes at one point in your biology career here at Conrad. So we're getting back into it, right? Now, if you see, right, you can go take a look at one of these individual guys, right? So I'll go ahead and try to get the color to match as best as I can. I was like, seeing, do I have black here? I don't even know if I have black. I don't think I do. Anyway, so you end up having this, the little circle, and then you end up having these little dangly tails right here, right? So this is a phospholipid right there. And what we can do is start to talk a little bit about why it orients itself right here, right? So first of all, let's talk about hydrophilic tail, right? Hydrophilic, uh, oh, that is actually incorrect. Whoa, we got to scribble that out. That is wrong. Why is it said hydrophilic? That's wrong. It's supposed to be hydrophobic. This is a bad picture, everybody. Hydrophobic. Hydrophobic. So hydrophobic tail. I just found out an issue with that. Yeah, scratch that guy off. That is wrong. That's a bad picture from the internet. Bad picture, internet. I gotta go change that for next year. All right. Anyway, so you're hydrophobic, right? Hydro, right? Hydrophobic. You also have hydrophilic, right? Hydro meaning water, right? So the philic, if you remember, right? Philic is going to be mentioning boop. This means that it is water liking, right? So the heads, right? So this head and this head, they like water, right? This one also likes water, right? Because if this is our cell, we have water here on the outside of the cell, and we have water located on the inside of the cell, right? So we're good with that. But then if we go and take a look at this middle area, I'll kind of do it in a different color, right? A little bit of like purple's not going to work. Let's Let's do like just a red or something. Make that simple. Okay, here, right, this is the hydrophobic region, right? Phobic means that this is water fearing or water hating, right? Fearing or water hate. And what that is that means that there's not going to be any water right here. So what happens is that the water is located on the outside of the cell and the inside of the cell, but we're not going to have any water here, or at least that's the intent behind it. When we start talking about diffusion and osmosis specifically, there's like a chance that a water molecule might be able like to pop on through from one side or another, but it's kind of hard for it to happen. And that's because it's all about the polarity, right? So let's go ahead and kind of scrub this for a second and talk through that, okay? So this is hydrophobic, right? This is the hydro, I'm just like getting upset. Hydrophobic, right? So this is hydrophobic, right? So this is non-polar, right? And also keep in mind, these are lipids, right? These are lipids. And what do you remember about lipids, right? They're generally nonpolar, right? So what we end up doing is that we take our triglyceride, right? We got that triglyceride, right? And what I end up doing is I lob off one of those fatty acid chains, and instead I throw a phosphate group over here, and that's what gives us this phosphate group. And now a phosphate group is super negative. It's like a phosphorus and all these oxygens over there. It's like super negative, right? Which means that I have a definitive negative side. So that means that this is going to be polar up here. Let me do that in a different color so that way we can kind of keep track of the stuff, right? So you're going to have polar up here and polar over there, right? And how things work, right? If you put water, right, and if you try to mix it, with oil, right, plus oil, we know that they don't mix. That this is not what ends up happening, right? So the water will want to interact with other water, while the oil or like the nonpolar stuff is only going to want to work with nonpolar. This is why it orients itself the way that it does. Like if I'm looking at the phospholipid bilayer, this area does not want to interact with this neighboring area. It's nonpolar here and it's polar over there. So again, right, nonpolar interacts with nonpolar, polar interacts with polar. That's why these guys orient themselves as like their little dangly bits are facing each other. That's those fatty acid layers. Okay. So this is just going into a little bit more detail about like literally everything we ended up talking about. If you need to write this down, where your heads are hydrophobic, right? They're polar. Sorry, the heads are hydrophilic. The heads are hydrophilic. They are polar, so they are going to want to go and face the water, which is why we see that it is facing the water over here, right? So these heads are facing the water here, and these heads are facing that water over there. While the tails, right, they are non-polar, right? They're non-polar, so they are trying to stay away from the water, right? And what this does is that it makes a definitive barrier, and that's kind of like the big thing about this, right? Here's the outside of the cell, and then here is the inside of the cell, right? So I'm actually having it's almost like your skin, right? You have a definitive way of being inside and outside. Now, what's really neat about these phospholipids is that, well, wait a second, can it actually make other shapes aside from the bilayer? And it can. There's other shapes that are out there, but the one that we really kind of just focus on are just like these cell membranes, right? Now, this is an image kind of showing you what this ends up looking like, right? So here's like the cell membrane, right? Again, cells like to be spheres or cylinders or cubes, right? We like to be small stuff, right? Maximizing that surface area to volume ratio. What you'd actually end up finding is that as you get bigger, you need more surface proteins in order to go and let things in and out. And it's another reason why it ends up becoming an issue. But that's neither here nor there. We don't need to worry about that at the moment right now. But what I want to get you to be aware of, right, is that this is a sphere, right? So it's a three-dimensional object. So these phospholipids wrap all the way around and they come back over to here. So we're just kind of like looking at a cross section of this guy right here. All right. This image right here is actually showing you a little bit more detail. Here is the phosphate head, and then there are the fatty acid tails. I got a sneeze coming. We'll see if it ends up happening. It's like the worst. Okay, now what's really cool is that you're going to go see these pictures of these embedded proteins that are going to go be in here, right? Now, what's cool about this, I want you to go back to your notes for a second, okay? And what I'd like for you to go do is look at your, specifically, I need you to go look at the tertiary. Look at the tertiary folding for a protein. And I just want you to go take a look at the ionic charges. I want you to go look at the hydrophobic ones, right? You didn't write them down, but I just want you to be like, oh, right, that there's ionic, there's hydrophobic interactions, disulfide bridges. They're not really relevant for here, but just go look at that real fast and just have that page on hand, so that way you kind of understand this next bit that I'm going to go talk about. Okay. So hopefully you got that. So you can kind of like thumb through, pause the video if you need to in order to go back. So right. So we keep on saying that the heads are polar, right? So this is polar, and because we got water out here, this is also going to be a polar region, and we can mirror it being polar, polar, and also polar over here. And hopefully you understand that, right? So what that means is that if this is polar here, this protein needs to be structured in a way that our R groups from our tertiary folding, well, you're going to have ionic interactions here, and you'll have hydrophilic interactions out here as well. So you're going to have both of these guys facing the outside as well as facing these uh, phosphate heads, right? Because again, like interacts with like. Your polar wants to play with polar, and then that's going to get us to the next little bit, which is going to be right that the non-polar likes to interact with the non-polar. I'm trying to write with my fingers, right? So since this region is non-polar, this region's also non-polar. That means that the portion of the protein that is inside of the lipid bilayer, the phospholipid bilayer, this region of the protein is going to have non-polar segments in there. So like those methyl groups that were out there, right? Because this non-polar is going to want to interact with other non-polar stuff. So what I'm trying to make you think about, right, is that this is a protein that's embedded into our cell membrane. It's actually designed in a way that the non-polar regions and that the polar regions, right? The polar regions are facing the correct way, right? They're facing the correct way. And then you can maybe start to think, well, what would happen, right? If let's say that I were to put something else in here, like maybe like I had like maybe like a non-polar substance in here. Well, that non-polar substance is not going to want to interact with the polar heads, but it might have an interaction with these uh, phospholipid tails, and then that can end up like reversing the whole membrane, and then it starts to deconstruct itself. This non-polar region will not interact with that, but it wants to interact with this region, so it can cause some really funky protein folding, right? So so what I'm getting at is like depending on the environment that you're in, it can definitely affect how these proteins work. And then also, we haven't gotten into it, but we'll eventually down in I'm going to just say for like unit 6, we're going to be talking about mutations, where there is a change in the DNA, which ultimately changes the shape of the protein. And now, if this protein doesn't have the correct polar and non-polar regions, it's not going to form properly, and then it won't be able to do its job, right? What was that mantra we kept on saying? Structure determines function, right? So if the shape of the protein doesn't work properly, if it cannot embed itself within the cell membrane correctly, then it's not going to be able to do its job. This could, whether it be an enzyme or it be a transport protein or something. There are certain cases where people might not have a transport protein that needs to get like some kind of ion from like one side to the other. And if it doesn't have the ability to do that, then you can't actually transport this ion across, and then that can cause like some health issues. So these proteins, like they are designed in a specific way to make sure that they slot in into the cell membrane and that they are designed to specifically do a particular job. We end up talking about this. So this is a side section, like a cross section of our cell membrane. We call this the fluid mosaic model. So the reason why we call it a fluid mosaic model is that your proteins are constantly kind of like moving around. What I mean by that is like this protein can like shift over to here. Again, this is a three-dimensional thing, so it can shift like not like outside of it, but like if you can imagine like we're pushing it into the screen, that it's going like further and further and further away, like down this area for the phospholipids. Maybe this one right here is like coming towards you, right? So it's fluid, so things are able to move around and stuff, and that's that. You know, that's that. Going back to what we're talking about with the proteins, right? This is actually kind of showing you a couple of different examples of proteins where we have glycoproteins. We can have a glycolipid right over to here. This guy right here is a transport protein. You can have channel proteins where are like consistently open and stuff, and all these guys do different functions. And you can maybe start to think about yourself, right? It's like, okay, so here is a polar region here, while it is non-polar over here, but then like maybe this area inside here is polar, so that way it can interact with the stuff outside there. And it's just like there's a design to these proteins, and if it's kind of going above your head, like ask questions about this. It's kind of going a little bit above and beyond, but it does help you understand like why we talked about like those IMFs and why those intermolecular forces are so incredibly important, and why like I harp on them so much at that. All right. Oh, so this is just telling you that the cell membrane is constantly moving, specifically the stuff that's within the membrane. So the structures are able to go float around, and even sometimes like the phospholipids, they can go from one side to another. And as long as they stay in the orientation, they're going to be perfectly okay. Right? These are a couple of the different guys that you'll go ahead and take a look at. So you have steroids, which are cholesterol right in here. So if we go and take a look at this, so these little tiny yellow globule things right here, that's going to be your steroids. That's your cholesterol. They're just there to help out like fluidity. In order to just make sure that the cell membrane like is able to be fluid as well as maintains its shape. Proteins, that's pretty much all these guys that are hanging out in here. Now we can like change the actual name of like what kind of protein it is by saying if it's like a transport protein or what have you and stuff like that. But you know, we'll get to that in a little bit. The next ones that you have is the glycoprotein and the glycolipid. What I'm going to go do is just kind of keep in mind, right? So glyco and glyco is exactly the same, right? So in the case here, your glyco protein will have some kind of carbohydrate, like usually associated with it. But then you got glycolipid, where it's attached to a lipid, and then the glycoprotein is just it's attached to the protein. So to get you a little bit familiar or aware of it, your glycolipid is this guy right here. You can kind of see how there's like a little phospholipid right here that it's attached to. It's just how this ends up being designed. So like it just kind of like slots itself into the cell membrane, and it works as a surface protein. In the case of a glycoprotein, in case of a glycoprotein, that's going to be like these guys right here. There's going to be some kind of carbohydrate chain that's out there, and they're used for cellular communication. So the purposes behind the cellular communication is just allows them to bump into each other and know where you are or know what kind of cell that you are. One of the most common examples is blood typing, and we'll go into more about blood typing in a little bit. Sort of like it like later on down like the curriculum, but I can kind of give you a little bit of a idea, right? So you may know that the different blood types is A, B, AB, and O. And for the sake of this, I'm just going to go talk about like A, right? So in the case here, what this means is that you have this A antigen, which is just a glycoprotein that's located on your cell membrane. So this is like this can be the guy that says like, hey, I am a blood type, right? And this is what actually ends up determining like, hey, I am blood type A, right? And then you know, and when we talk about antibodies and stuff like that, you like the people who have the A blood, you'll have the B antibodies. I'm kind of going a little bit off script, but just kind of follow me for a second, right? They're A antibodies, right? Sorry, B antibodies. So these B antibodies are looking for the B antigen, but this cell has, or this body has the A antigen, so it's not going to get affected by it. But let's say that I were to give somebody, right? I gave my A blood to somebody who had like the B blood. I'll do this in a different color so you can kind of like keep track of what's happening, right? If I did this like with the B blood right here, and maybe this was actually a B, and we'll say that this didn't affect it, well, this antibody would attack that antigen, and that can be used for that's like cellular communication, right? Allow it to realize like, hey, this is like something that's foreign to my body. I'm going to go ahead and attack it. Back in the day, before we knew about blood typing, you would go and do a blood transfusion. It would kill the person. Like, we don't understand why. We understand that blood is like what's going to keep you alive. And they found out that basically what happened was that your blood would agglutinate or clump up, and then it would cause like a clot somewhere. And if you can't push the clot through it, your heart or your brain suffocates and it's unable to do its job, and you perish from that. So that's like a lot of information just as an introduction to the plasma membrane and the cell membrane. So what we'll do is we'll go ahead and pick it back up, and we'll talk through a little bit about like the movement on how stuff goes from one area to one side to another. So that'd be it, everybody. Thank you.